Dear students, I am Sanjeev Singh, Assistant Professor GLA College, and I was discussing DNA repair. I have already discussed one mechanism, and this topic belongs to BSc Semester 3 Botany, NPU University. So the second method of DNA repair is nucleotide excision repair. Now, in earlier when we discussed base excision repair, the base was excised first. Now the nucleotide will be excised. Nucleotide means the sugar, the phosphate and the base, all three included. Now the damaged nucleotides are excised, which means like the dimer which is induced by UV light or some other factors. Now dimer is a structure which, when generally it is the thymine, two thymines of adjacent thymines in one strand, they tend to join together and form a dimer. So when the new strand is being formed, proper pairing does not take place. So this dimer needs to be removed. For example, in E. coli, how it is removed? There are different proteins which are involved in this process. In E. coli, the proteins are UVRA, UVRB and UVRC. These three proteins, they get together and they remove the damaged nucleotide. The damaged nucleotide are removed by this strand. Fine. Removes by this, the damaged nucleotide are removed by these three proteins. Now when the nucleotide is removed, the gap is created, and now we all know that the gap is to be filled, which is filled by DNA polymerase 1, which is the enzyme responsible for synthesis of the DNA, and then the ends are joined by DNA ligase. Now, in different prokaryotes, the UVRs, the proteins which are used, they are named differently. For example, in yeast, it is named as RADX. RAD stands for radiation. It is termed as RAD3, RAD10, etc. So now we will try to understand this in the diagram, how it happens. So first you see a DNA sequence, and you see the damaged nucleotide over here, the dimers. These two are joined together, so the pairing does not happen. Now this damaged nucleotide is removed by the combination of this UVARA, UVRB and UVRC. You see it has bound here. It has bound here. Fine. Now after that protein binds, those combination of three protein binds, the nucleotide is excised from this place and this place. So gap is created over here. And this gap is then filled by DNA polymerase 1 and DNA ligase. And the repair is complete. And now in the next video, I will discuss the mismatch repair.